Hey there, science fans! Today we're diving into the fascinating world of the cell cycle and mitosis. It's all about how cells grow and divide, which is like their own circle of life. This isn't just some abstract biology concept, though. For all you NEET UG aspirants out there, understanding cell division is crucial for acing your exams. Think about it. Growth, repair, reproduction, almost everything in biology boils down to cells dividing properly. So buckle up, because mastering this topic will give you a big advantage on the big day. Let's get started. Before a cell can divide, it needs to grow and duplicate its insides. That's where interphase comes in, the longest part of the cell cycle. Interphase is like the preparation stage before a big race. It has three main phases, G1, S, and G2. In G1, the cell is growing like crazy, making more proteins and organelles. Think of it as bulking up for the big event. Next up is the S phase, where DNA replication steals the show. The S stands for synthesis, and that's exactly what happens to the DNA. It gets copied. Imagine each strand of DNA as a zipper. During the S phase, the zipper unzips and each side gets a new matching side. Now we have two identical copies of the DNA. Finally, in G2, the cell double-checks everything. It's like a final quality control step before the grand finale of mitosis. Get ready for the main event, mitosis. It's time for the cell to divide its nucleus and DNA, ensuring each new cell gets a complete set of instructions. Prophase kicks things off with a flurry of activity. First, the long, stringy DNA molecules condense into compact structures called chromosomes. And imagine trying to move a giant bowl of spaghetti. It would be much easier if the spaghetti was bundled up neatly. As the chromosomes condense, they become visible under a microscope. You'll notice that each chromosome has two identical copies called sister chromatids, attached at a point called the centromere. Meanwhile, the nuclear membrane, which normally surrounds the DNA, starts to break down. It's like removing the curtain before a big reveal. And finally, the mitotic spindle, made of microtubules, starts to form. These microtubules act like tiny ropes that will eventually pull the chromosomes apart. With the stage set, it's time for metaphase, the shortest but most dramatic part of mitosis. During metaphase, the chromosomes perform a remarkable act of synchronization. They line up neatly along the cell's equator, also known as the metaphase plate. Imagine a perfectly choreographed dance where each chromosome knows exactly where to stand. The mitotic spindle fibers, which we met in Prophase, play a crucial role here. They attach to the centromeres of the chromosomes, ensuring that each sister chromatid is connected to opposite poles of the cell. This alignment is crucial for ensuring that each new cell receives a complete set of chromosomes. It's like making sure each team in a tug-of-war has an equal number of players, eh? Section 5, anaphase, pulling chromosomes apart. Hold on tight, because things are about to get even more exciting. Anaphase is when the real action begins. The sister chromatids are ready to part ways. Remember those spindle fibers attached to the chromosomes? In anaphase, they shorten, pulling the sister chromatids apart towards opposite poles of the cell. Imagine a perfectly synchronized tug of war where both teams pull with equal force, causing the rope to split in half. As the sister chromatids separate, they are now considered individual chromosomes. They move rapidly towards the poles, ensuring that each new cell will receive a complete set of genetic information. Section 6, telophase 2, new nuclei, take shape. After the dramatic separation of chromosomes in anaphase, telophase brings a sense of calm and order as the cell begins to divide into two distinct daughter cells. The separated chromosomes, having reached their respective poles, start to uncoil and become less condensed. They are no longer needed in their tightly packed form as the cell is preparing to divide. Meanwhile, the nuclear envelope, which had broken down in prophase, reforms around each set of chromosomes. It's like putting the curtain back up after the big show. Two new nuclei are now formed, each containing a complete set of chromosomes. As the nuclei settle into their new homes, the mitotic spindle, which played such a crucial role in chromosome separation, disassembles. Its job is done, and the cell is almost ready for its final split. Section 7, cytokinesis, dividing the cytoplasm. We've successfully divided the nucleus, but what about the rest of the cell? That's where cytokinesis comes in, the final act of cell division. Cytokinesis is like drawing a line in the sand, dividing the cytoplasm and organelles equally between the two new daughter cells. In animal cells, a cleavage furrow forms, pinching the cell membrane inward until it eventually splits into two. Imagine squeezing a balloon in the middle. That's similar to how the cleavage furrow works. Plant cells, with their rigid cell walls, can't divide in the same way. Instead, they form a cell plate in the middle, which gradually expands outwards until it fuses with the existing cell wall, creating two separate cells. And there you have it, two identical daughter cells, each with its own nucleus, DNA, and a full set of organelles, ready to start the cell cycle all over again. Section 9, NEET, UG, Connection, Mastering Cell Division for your exam. Now that we've explored the fascinating world of the cell cycle and mitosis, let's talk about why it's so important for your NEET UG exam. Questions related to cell division are a staple in the NEET UG biology section, and for good reason. This topic is fundamental to understanding a wide range of biological processes, from growth and development to genetics and evolution. Expect questions that test your knowledge of the different stages of the cell cycle, the key events that occur during each stage of mitosis, and the differences between mitosis in plant and animal cells. You might be asked to identify different stages of mitosis from microscopic images, explain the significance of specific events like DNA replication or chromosome separation, or even predict the consequences of errors during cell division. The key to mastering this topic is to develop a clear understanding of the underlying concepts rather than simply memorizing facts. Focus on visualizing the processes, understanding the reasons behind each step, and relating the information to real-world examples. By mastering the cell cycle and mitosis, you'll be well equipped to tackle those NEET UG biology questions with confidence. Section 10. Conclusion. The never-ending dance of cell division. As we conclude our journey through the cell cycle and mitosis, let's take a moment to appreciate the incredible importance of this process in the grand scheme of life. From the moment we are conceived to our very last breath, our cells are constantly dividing, replacing old or damaged cells, ensuring the growth and maintenance of our bodies. It's a never-ending dance, a cycle of growth, division, and renewal that underpins the very essence of life. By understanding the intricacies of the cell cycle and mitosis, we gain a deeper appreciation for the remarkable complexity and elegance of life, from the smallest single-celled organisms to the largest and most complex creatures on Earth. So keep exploring, keep asking questions, and keep marveling at the wonders of the microscopic world, because within each tiny cell lies the secret to life itself.
Section 1. The grand stage of life, introducing meiosis. Life is a grand play, and at the heart of this production lies a fundamental process known as cell division. This process ensures the continuity of life, allowing organisms to grow, repair, and reproduce. Meiosis, a specialized form of cell division, takes center stage in the drama of sexual reproduction. It is the driving force behind the incredible diversity we see in the living world. Imagine a world without variation. Every human being would be identical, every rose would look the same, and the richness of life's tapestry would be lost. Meiosis, through its intricate dance of chromosomes, ensures that this is not our reality. It shuffles the genetic deck, creating unique combinations of genes that are passed down from one generation to the next. This essay delves into the captivating world of meiosis, uncovering its secrets and highlighting its significance for NEET UG aspirants. We will embark on a journey through the microscopic world of chromosomes, exploring the key events and structures involved in this vital process. Get ready to unravel the mysteries of meiosis and understand why it is considered a cornerstone of genetics and evolution. Section 2. The two-act play. An overview of meiosis. Meiosis, often referred to as reduction division, is a type of cell division that reduces the number of chromosomes in a parent cell by half, resulting in the formation of 4 haploid daughter cells. These haploid cells, containing only one set of chromosomes, are the gametes, sperm and egg cells, that unite during fertilization to create a new organism. The meiotic dance is beautifully choreographed into 2 major acts: Meiosis I and Meiosis II. Think of Meiosis I as the separation of the dance partners, where homologous chromosomes, one inherited from each parent, part ways. This division ensures that each daughter cell receives a complete set of genetic information, albeit a mixture of maternal and paternal origin. Meiosis II, on the other hand, focuses on the separation of sister chromatids, identical copies of a chromosome that are attached at a region called the centromere. This second division ensures that each of the 4 daughter cells receives only one copy of each chromosome, maintaining the correct chromosome number across generations. Section 3. Act 1. Meiosis I. Separating the dancers. Meiosis I, the first act of our meiotic play, is where the magic of genetic recombination unfolds. It is during this stage that homologous chromosomes pair up, exchange genetic material, and then bid each other farewell. Let's take a closer look at the 4 scenes that make up this captivating act.
Scene 1. Prophase 1. Pairing up and switching steps. The curtain rises on Prophase 1, the longest and arguably the most eventful stage of meiosis. Here, the chromatin, the diffuse form of DNA, condenses into visible chromosomes, each consisting of 2 sister chromatids joined at the centromere. A crucial event called synapsis then takes place, where homologous chromosomes pair up, forming a structure called a tetrad, or bivalent. Imagine 2 dancers, each representing a homologous chromosome, coming together and intertwining their arms. This close association allows for an extraordinary exchange of genetic material between non-sister chromatids in a process known as crossing over. Like dancers switching steps, crossing over results in new combinations of genes on the chromosomes, increasing genetic diversity.
As Prophase 1 draws to a close, the nuclear envelope breaks down and the stage is set for Metaphase 1. The spotlight now shines on the tetrads as they line up along the cell's equator, or metaphase plate. Microtubules emanating from opposite poles of the cell, attached to the centromeres of the homologous chromosomes, ready to pull them apart.
With a dramatic flourish, anaphase commences, and the homologous chromosomes, each still composed of 2 sister chromatids, are drawn apart by the spindle fibers. Imagine the dance partners now separating and moving towards opposite ends of the stage. This separation marks the first step towards the formation of haploid cells.
As the first act of our meiotic play comes to a close, telophase unfolds. The separated chromosomes reach the poles of the cell, and the nuclear envelope may reappear. The cytoplasm divides in a process called cytokinesis, resulting in the formation of 2 daughter cells, each with half the number of chromosomes as the original parent cell. Think of this as the intermission, a brief pause before the second act begins. Each daughter cell now contains a haploid set of chromosomes, but each chromosome still consists of 2 sister chromatids.
Section 4, Act 2, Meiosis II, the spotlight on sister chromatids. As the curtain rises on Act 2, the spotlight shifts to the sister chromatids, the identical twins that have remained together until now. Meiosis II closely resembles the events of mitosis, the process of cell division that occurs in somatic cells. However, unlike mitosis, which starts with a diploid cell, meiosis II begins with two haploid cells produced in meiosis I.
Scene 1. Prophase 2. A brief pause. Prophase 2 is a relatively short and uneventful scene compared to its counterpart in meiosis I. The chromosomes condense again, becoming visible under the microscope. The nuclear envelope, if it had reformed during telophase I, breaks down and the spindle fibers begin to assemble. Scene 2. Metaphase 2. Aligning once more. In metaphase 2, the chromosomes line up single file along the metaphase plate. Their centromeres attached to spindle fibers extending from opposite poles. This alignment is crucial for the equal distribution of genetic material to the daughter cells. The separation of the sister chromatids marks the climax of anaphase II. The centromeres divide, and the sister chromatids, now considered individual chromosomes, are pulled apart by the spindle fibers towards opposite poles of the cell. Imagine the final separation of the dance partners, each now embarking on a solo journey. As Act 2 draws to a close, telophase 2 unfolds. The chromosomes arrive at the poles and the nuclear envelope reforms around each set of chromosomes. Cytokinesis follows, dividing the cytoplasm and resulting in the formation of 4 haploid daughter cells, each genetically unique. The final curtain falls on our meiotic play, leaving behind a legacy of genetic diversity. The 4 haploid cells, each carrying a unique combination of genes, are the culmination of this intricate dance of chromosomes.
Meiosis is not merely about reducing the number of chromosomes, it's about shuffling the genetic deck, ensuring that each gamete is a unique blend of parental genes. This genetic variation is the raw material upon which natural selection acts, driving evolution and shaping the diversity of life we see around us. 2 key events during meiosis contribute significantly to this genetic shuffling: crossing over. The exchange of genetic material between non-sister chromatids during prophase I creates new combinations of alleles or different versions of a gene on the chromosomes. This process ensures that even genes located on the same chromosome can be inherited independently, further increasing genetic diversity. Independent assortment. During metaphase I, the random alignment of homologous chromosomes at the equator of the cell leads to different combinations of maternal and paternal chromosomes in the daughter cells. This random assortment of chromosomes, like shuffling a deck of cards, generates a vast number of possible gametes, each with a unique genetic makeup. This remarkable ability of meiosis to generate genetic variation is central to the process of evolution. It provides the raw material upon which natural selection can act, favoring individuals with traits that enhance their survival and reproduction in a given environment. Understanding meiosis is not just an academic exercise, it's crucial for NEET UG aspirants aiming to excel in the biology section. This intricate process forms the foundation of genetics, inheritance, and evolution, all of which are high-yield topics in the NEET UG exam. Here's how a thorough understanding of meiosis can benefit you. Conceptual clarity. Meiosis is a complex process with multiple stages and intricate events. Mastering this topic will strengthen your grasp of fundamental biological concepts, making it easier to tackle related topics in genetics and evolution. Problem-solving skills. NEET UG. Questions often involve applying your knowledge of meiosis to solve problems related to inheritance patterns, genetic disorders, and chromosomal abnormalities. A strong foundation in meiosis will equip you with the problem-solving skills needed to excel in these areas. Application to real-world scenarios. Understanding meiosis is not just about memorizing facts, it's about applying your knowledge to real-world scenarios. For instance, knowing how non-disjunction during meiosis can lead to genetic disorders like Down syndrome highlights the clinical significance of this process. As you embark on your NEET UG preparation journey, remember that mastering meiosis is not about rote memorization, but about building a deep understanding of the process and its implications. Embrace the visual nature of this topic, utilizing diagrams and animations to solidify your understanding of the different stages and events. Practice makes perfect, so make sure to work through past NEET UG questions related to meiosis. Refer to your NCERT textbooks for a comprehensive understanding of the topic, paying close attention to diagrams, tables, and key concepts. As you delve deeper into the world of meiosis, you'll discover the elegance and complexity of this fundamental biological process. This knowledge will not only equip you to excel in your NEET UG exams, but also provide you with a deeper appreciation for the wonders of life and the intricate mechanisms that drive its diversity.
Imagine a vast, intricate dance floor, where countless dancers twirl and sway in perfect harmony. Each movement is precise, each step choreographed to perfection, creating a mesmerizing spectacle of life in motion. This is the world inside your body, a world teeming with trillions of cells, each engaged in a delicate ballet of life. These cells are not just passive entities, they are dynamic, constantly communicating, adapting, and responding to their environment. Cells are the fundamental units of all living things, constantly dividing and multiplying to replace old or damaged comrades and ensure the survival of the organism they compose. This relentless cycle of birth, growth, and renewal is what keeps us alive and thriving. This elegant process of cell division, essential for growth, repair, and reproduction, comes in two main forms: mitosis and meiosis. Each type of division has its own unique role and significance in the grand scheme of life. Mitosis, the more common of the two, is akin to a cloning dance. It is the process by which a single cell divides to produce two identical daughter cells, each a perfect copy of the original. One cell, called the parent cell, duplicates its genetic material and then gracefully divides into two identical daughter cells. This process ensures that each new cell has the same genetic information as the parent cell, maintaining the continuity of life. These daughter cells are exact replicas of the parent, ensuring genetic continuity from one generation to the next. This precise duplication is crucial for the growth and maintenance of tissues and organs. They carry the same blueprint, the same instructions for life. This genetic blueprint, encoded in the DNA, dictates everything from the cell's structure to its function, ensuring that each cell performs its role effectively. Meiosis, on the other hand, is a more specialized form of cell division, reserved for the creation of gametes, sperm, and egg cells. This process is essential for sexual reproduction, allowing for the mixing of genetic material from two parents. Here, the dance takes on a different rhythm, resulting in 4 daughter cells, each with half the genetic material of the parent cell. This reduction in genetic material is crucial for maintaining the correct number of chromosomes in offspring. This reduction in genetic material is crucial for sexual reproduction, allowing for the combination of genetic information from two parents. When a sperm cell and an egg cell unite during fertilization, they create a new organism with a complete set of chromosomes. Whether it's the cloning dance of mitosis or the intricate choreography of meiosis, cell division is a tightly regulated process, orchestrated by a complex network of proteins and checkpoints. These regulatory mechanisms ensure that each step of the division process is carried out accurately and efficiently. These checkpoints act like strict quality control inspectors, ensuring that each step of the division process is carried out flawlessly before the next begins. They monitor the cell's progress, checking for errors and making corrections as needed to prevent the propagation of damaged or incomplete cells. When these checkpoints fail, the consequences can be dire. Errors in cell division can lead to the development of cancer and other diseases, highlighting the importance of these regulatory mechanisms in maintaining cellular health and function. Despite the complexity and potential for error, the process of cell division is remarkably efficient and reliable. It is a testament to the incredible precision and coordination of the cellular machinery that keeps us alive and well. So the next time you think about the trillions of cells in your body, remember the intricate dance they perform every day. It is a dance of life, a dance that sustains us, heals us, and connects us to the very essence of existence. Imagine a bustling checkpoint at a border crossing, where the flow of people and vehicles is constant, and the atmosphere is charged with anticipation and vigilance. Here, guards meticulously scrutinize passports and visas before granting entry, ensuring that only those who meet the necessary criteria are allowed to pass through. This scenario is akin to the G1/S checkpoint in the cell cycle, the first critical juncture that a cell encounters. It is situated at the transition between the G1 phase, a period dedicated to cell growth and preparation, and the S phase, where the essential process of DNA replication occurs. At this checkpoint, the cell faces a crucial decision to divide or not to divide. This decision is not taken lightly, as it determines the cell's future and its ability to contribute to the organism's growth and repair. The answer hinges on a variety of factors, including the availability of resources, the presence of growth signals, and the integrity of the cell's DNA. These factors collectively ensure that the cell is in optimal condition to proceed with division. Like vigilant border patrol agents, specialized proteins within the cell monitor these conditions. They act as sentinels, ensuring that the cell is ready for the momentous task of DNA replication, and that all necessary criteria are met. One of the most important proteins involved in this first checkpoint is p53, often referred to as the guardian of the genome. This protein plays a pivotal role in maintaining the integrity of the cell's genetic material. This protein acts like a meticulous inspector, carefully scrutinizing the cell's DNA for any signs of damage. It ensures that the genetic information is intact and free from errors that could be detrimental to the cell's function. If p53 detects any abnormalities, it can halt the cell cycle, preventing the damaged DNA from being replicated. This pause allows the cell time to repair the damage, ensuring that only healthy DNA is passed on to the next generation of cells. Think of p53 as a meticulous mechanic inspecting a car before a long journey. Just as a mechanic ensures that a vehicle is in perfect condition before it hits the road, p53 ensures that the cell's DNA is in perfect condition before it proceeds with division. If any significant issues are found, the mechanic won't let the car leave the garage until the problems are fixed. Similarly, p53 will not allow the cell to proceed with division until any DNA damage is repaired. Similarly, p53 ensures that only cells with healthy DNA are cleared to proceed to the next stage of the cell cycle. This checkpoint acts as a quality control mechanism, safeguarding the cell's genetic integrity. The G1/S checkpoint is a critical safeguard against uncontrolled cell growth and the development of diseases like cancer. By preventing cells with damaged DNA from dividing, it helps to maintain the overall health of the organism. By ensuring that only healthy cells with intact DNA proceed to division, this checkpoint plays a vital role in maintaining the integrity of our genetic information. It acts as a gatekeeper, allowing only the best prepared cells to continue their journey through the cell cycle, thereby preserving the health and stability of the organism as a whole. When the G1/S checkpoint fails, the consequences can be dire. Unchecked cell division can lead to the formation of tumors and the development of cancer. This highlights the importance of this checkpoint in protecting the organism from such devastating outcomes. In a healthy cell cycle, all checkpoints, including the G1/S checkpoint, function properly to ensure that cell division occurs in a controlled and regulated manner. This meticulous regulation is essential for the maintenance of healthy tissues and the prevention of diseases. When a cell successfully passes through the G1/S checkpoint, it signifies that the cell is ready to replicate its DNA and proceed with division. This successful transition is a testament to the cell's preparedness and the effectiveness of the checkpoint mechanisms in place. The G1/S checkpoint, along with other checkpoints in the cell cycle, plays a crucial role in ensuring the fidelity of cell division. These checkpoints act as guardians, protecting the organism from potential genetic errors and maintaining the overall health of the cells. By maintaining strict control over the cell cycle, these checkpoints contribute to the overall health and stability of the organism. They ensure that cell division occurs in a precise and orderly manner, preventing the development of diseases and promoting the well-being of the organism. In conclusion, the G1/S checkpoint is a vital component of the cell cycle, acting as a gatekeeper that ensures only healthy cells with intact DNA proceed to division. Its role in maintaining genetic integrity and preventing uncontrolled cell growth underscores its importance in the overall health of the organism. By understanding the function and significance of the G1/S checkpoint, we gain insight into the intricate mechanisms that govern cell division and the maintenance of genetic integrity. This knowledge is crucial for advancing our understanding of cellular processes and developing strategies to combat diseases like cancer. Ultimately, the G1/S checkpoint exemplifies the complexity and precision of cellular regulation, highlighting the importance of maintaining strict control over the cell cycle to ensure the health and stability of the organism. Imagine a team of editors meticulously proofreading a manuscript before it goes to print, ensuring every sentence is grammatically sound and factually accurate. They scrutinize every word, every punctuation mark, and every fact, making sure that the final product is polished and error-free. This process is crucial because even a small mistake can change the meaning of a sentence or mislead the reader. This is the role of the G2/M checkpoint, the second major checkpoint in the cell cycle, stationed at the transition between the G2 phase, where the cell prepares for mitosis, and the M phase, where the cell physically divides. Just like the editors, this checkpoint ensures that everything is in order before the cell proceeds to the next stage. This checkpoint serves as a critical quality control step, ensuring that DNA replication has been completed accurately and the cell is adequately prepared for the complex choreography of mitosis. During the G2 phase, the cell has already duplicated its DNA, and now it must ensure that this duplication process was flawless. Just as a manuscript with errors can lead to confusion and misinterpretations, errors in DNA replication can have dire consequences for the cell and the organism as a whole. These errors can result in mutations, which might lead to malfunctioning proteins or even cancer. At the G2/M checkpoint, the cell confirms that its DNA has been fully replicated and that there are no lingering errors or damage. This involves a thorough inspection of the newly synthesized DNA strands to ensure their integrity. It also checks to ensure that the cell has synthesized enough proteins and organelles to equip both daughter cells with the machinery they need to function properly. This includes verifying that all necessary components are present and correctly assembled. If any problems are detected at this stage, the cell cycle is temporarily halted, giving the cell time to make repairs or adjustments. This pause is essential because it prevents the cell from dividing with errors that could be passed on to the daughter cells. This might involve activating DNA repair mechanisms to fix any errors in the newly synthesized DNA strands, or delaying entry into mitosis until the cell has accumulated sufficient resources. The cell has a variety of repair pathways at its disposal, each tailored to fix specific types of DNA damage. The G2/M checkpoint acts like a final dress rehearsal before the main performance of mitosis. It ensures that all the preparations are complete and that the cell is ready to divide. By ensuring that all systems are go and the stage is set for a flawless division, this checkpoint plays a crucial role in preventing errors that could lead to genetic instability and disease. The meticulous checks and balances at this stage are vital for maintaining the health and stability of the organism. The G2/M checkpoint is just one part of a larger network of regulatory mechanisms that govern the cell cycle. Each checkpoint serves a unique purpose, but they all work together to ensure that cell division proceeds smoothly and accurately. Just as a well-edited manuscript can inform and educate, a properly regulated cell cycle can lead to healthy growth and development. Conversely, errors in either process can have serious consequences, highlighting the importance of these checkpoints in maintaining order and accuracy. The seamless transition between the G2 and M phases, facilitated by the G2/M checkpoint, exemplifies the precision and coordination required for successful cell division. This checkpoint is a testament to the cell's ability to self-regulate and ensure its own survival. In the end, just as a successfully published manuscript is the result of careful editing and review, a healthy cell division is the outcome of rigorous checkpoints and quality control. The G2/M checkpoint is a crucial part of this process, safeguarding the integrity of the cell's genetic material and ensuring the proper distribution of resources to the daughter cells.